Hi, welcome everybody. I'm so glad to see the rain has not held anybody back from attending today. And I'm really delighted to welcome you to Quinnipiac University's North Haven campus. This is the home of our School of Medicine, School of Nursing, School of Health Sciences, our School of Education, and our School of Law. Just 9 minutes down the road is where our main campus is, our Mount Carmel campus, where the College of Arts and Sciences is located, and within which our behavioral neuroscience program and our psychology program are also housed. And just a quarter mile from there is our York Hill campus. I hope at some point, not on a rainy day like this, you'll get a chance to experience all of that. As Dean of the College of Arts and Sciences, I'm so happy, proud, and privileged to be able to sponsor the NEURON Conference and neuroscience in general, because it's such a remarkable, groundbreaking, and integrated field of study that really represents the best of what the College of Arts and Sciences has to offer. And I want to especially thank our co-sponsors today, the University of Connecticut, and in particular, the organizers from both institutions and our student helpers as well. From UConn, Jeff Tanner, Aleph Echebitoglu, that's a hard one, Elizabeth Klein, also from Yale as well, Jen Haoyang. From Quinnipiac, our faculty, Adrian Betts, Josh Tate, Professor Todd Ahern, and also Victor Van Cohn from our School of Medicine, our Quinnipiac and University of Connecticut student helpers, and many people who are behind the scenes who are supporting the conference today as well, including our administrative support professionals like Alex Lucia and Kathy Martin, our Associate Dean for Administration and Finance, Pat Wali, who's sitting in the audience today, and to the School of Medicine in general and the Dean of the School of Medicine, Phil Boissel, for sharing this beautiful facility with all of us so that you can experience what it's like to be in a School of Medicine building. I know from the excitement I see on all of your faces from the amazing program that's been put together that this is going to be an intellectually stimulating, a professionally rewarding, and I hope personally satisfying experience for everybody. And I hope it's an opportunity for you to really forge new connections, to share some of your knowledge and to learn from others, and to engage in the free and open exchange of ideas, which is exactly what conferences like this are intended to provide. So I wish you all a wonderful conference and experience here at Quinnipiac University's North Haven campus. I thank you for being here. I look forward to our incredible keynote address, and thank you very much, Professor Ahern.
Thank you, Adam. Also, thank you, Pat. Thank you for being here. So I'm going to run down a few of the logistics for today. And so first of all, I also want to extend my welcome to all of you for this, the 36th annual NEURON Conference. So pretty exciting. And I want to thank both Quinnipiac University and UConn for being co-sponsors here. We have a number of people to thank. Not everyone could fit on here, but in particular, we have our deans, we have faculty, we have the directors of various programs, particularly at UConn and Quinnipiac University. And so thank you all for your generous support of this amazing conference. This really is kind of a premier conference here in the Northeast, particularly for undergraduates. And so it would not be what it is without all of you showing up. So we're delighted to see you all here. We look forward to seeing your posters, and we're very excited about our keynote. Just so you can see our representation. So this was pulled from Eventbrite, and so you see we have a lot of representation from throughout the Northeast, all the way up into Maine and all the way south into New Jersey and so forth. And as you know, academics, we tend to wait till the last minute to get things done. So thank you all for registering. I really appreciate it. You can see that right there at the end, we love data and we love what it tells us about ourselves. So for those of you that want to follow along today, we do have the printed handouts, but there's a lot of information in there as well. That's amazing. So we also have a QR code on like the third or fourth page, maybe the second page, where you can get to the interactive program, and therefore you can search for the posters that might be of interest to you. It's also where we add in some last minute things. So there are a few extra posters that were added after we had to send the program to printing. So if you don't see your poster in there, you will only add it's online. We'll also add in people who helped us out today, just so that we have a full representation. So that is there at the bottom on the electronic version. Basically at the bottom of each section, you can click on the return to schedule or return to the table of contents, and that should allow you to navigate more easily. In terms of the schedule itself, you should find a page like this, and we are here. All right, so we are in the welcome and introduction phase. We're soon going to have the keynote, and I'm going to walk you through a few of the workshop options in just a minute. If you're interested in this conference, this obviously is the 36th year that we've had it, and we've been able to record a number of the previous keynote speakers. And so we'd encourage you to check out the Quinnipiac YouTube page. Make a little plug here for the Quinnipiac hockey team, which was just the champions in the country this year, so that was kind of exciting. Hey, but also, NEURON will probably be up there at some point too, so make sure that you For instance, last year we had Gerard speak to us about ketamine and the long journey from basically exploration into a clinical setting. And we're very excited this year to have Rebecca Shansky, who will be introduced in just a minute. Right. The workshops, what you can see here, we'll break out for you, is that we have a number of different workshops, and we tried to run a number of them twice so that you didn't have to choose between one or the other. You could choose one in the first session, and then you could choose a different one in the second or third session. You'll also see there that lunch is going to be in the second and third blocks, and that's going to be in the cafeteria. We should have signs, and you can find student helpers who can direct you in the right direction. For block number one here, this is going to start around 11:30, and you can see here that we have effective resume writing workshop, and that's our very own Quinnipiac College of Arts and Sciences Director of Career Development, Victor Vecchio. And we also have detectives of undiagnosed diseases, and then also for faculty, we have a workshop on kind of managing the context and conflicts that show up in labs. For block two, we have here an operative neuromonitoring and surgical neurophysiology. That will appear in block two and then also block three. And we again have the detectives of undiagnosed disease, and as we mentioned, lunch down in the cafeteria. We also have our data list, which will occur here in this auditorium, and we have a number of fantastic both graduate and undergraduate speakers lined up today. So we highly encourage you to swing by. And I just want to make a general announcement that we want you to explore these workshops, so don't feel stuck. If you want to move to another workshop because the workshop you decided on at first isn't quite the right fit for you, we encourage you to do that. We really want this day to be in support of your interests. Block three, we're going to have another data blitz as well as the resume writing workshop again, which showed up in block one, and then again the intraoperative neuromonitoring. Here's the list of the second set of data blitzes. Again, this is in your program if you have any questions. All right. After the workshops, we will have our undergraduate and graduate poster sessions. We had about 25 and more posters than we had last year. So we had to scramble to find room, so fantastic work. And you can see here that they're in sessions A through D. B and D are going to be upstairs on the second floor. A and C are going to be around the corner in the main sort of multi-purpose room. We'll then have a time where the judges will be able to judge the posters, and you'll be able to just kind of hang out. We'll have pizza delivered later this afternoon and some other light refreshments. The very end of the day, we have our poster awards, and so we encourage you to stay through all the way to the end. You do need to be present if you win an award. We also have a faculty award and our NeuroSci award, so be sure to stick around for that. More details are in the program. And finally here, it's kind of fun. It was a big success last year, but we have our photo booth out here, and so we encourage you to get your photo taken, and then if you tweet, we will go ahead and retweet it. So definitely take part in that. And I'm going to embarrass her, but I just noticed the president of the university, President Olian, is here, so I just want to welcome her. Thank you for making the trek over. And so I'm going to turn it now over to Professor Haight, another member of our behavioral neuroscience program here at QU, to introduce our speaker.
All right, welcome everyone. Thank you for everyone so far who's spoken. This has been a very warm welcome so far. So my job today is to introduce Dr. Rebecca Shansky. So Dr. Shansky is an associate professor in the Department of Psychology in the College of Science at Northeastern University in Boston. Dr. Shansky earned her PhD in neurobiology from Yale University here in New Haven, so welcome home. She studied sex differences and prefrontal cortical dysfunction in response to stress with Dr. Amy Arnstein while as a graduate student. Dr. Shansky continued her training as a postdoctoral fellow with Dr. John Morrison at Mount Sinai School of Medicine down in New York. Dr. Shansky then opened her own laboratory, the Laboratory of Neuroanatomy and Behavior at Northeastern in 2011. Her team uses rodent models to explore the neural connections between the medial prefrontal cortex and the amygdala, sex differences in how this circuit processes fear in response to stress, and how interactions between ovarian hormones and neurotransmitter systems influence this circuit. In addition to her leading research program, Dr. Shansky is a vocal advocate for gender and sex equity in experimental design. Highlighted recently is her Science Magazine Perspective piece, Are Hormones a Female Problem for Animal Research? Please join me in welcoming Dr. Rebecca Shansky today for her talk, What Have We Been Missing? New Insights into Pavlovian Fear Conditioning from Careful Consideration of Sex as a Biological Therapy.
Can everyone see okay? Or do you want the lights down? Does everyone see all right? Down a little bit? Okay. Yeah, so go ahead and do those. All right.
Thanks, yeah, perfect. All right, oh yeah, we got like moonlighting now. Thank you so much, everyone, for this warm welcome and for the invitation to speak. I'm really honored and really excited to share with you what's been going on in the lab recently. All right, so in the 80s, many of you don't remember this, but the makers of Reese's Peanut Butter Cups taught us that when events coincide in time, they become associated in our minds forever. And so this is essentially what those of us within behavioral neuroscience who are interested in learning and memory want to understand the mechanisms of how that happens within the brain. And so some of the questions that we're asking in the lab and across many labs all over the world is essentially, how does the brain form these associations? When things happen, the experiences that you have, you start to associate the things together. But for us as behavioral neuroscientists studying these processes in animals, we need some way of knowing whether the animal has actually formed the association, and because we can't simply ask it to remember when this thing happened, we have to trust our interpretation of the animal's behavior. And so the way that we study this in lab is through classical Pavlovian fear conditioning, which I assume most of you have seen before, been introduced before, but just to make sure everyone is on the same page, we take a rat, we put it in a box, we play an auditory tone that doesn't really have any kind of meaning or valence, it's just a noise. And when the tone ends, the animal gets a little foot shocks through the grid floor. And so those events have now coincided in time, and the animal starts to form an association between that conditioned stimulus and the unconditioned stimulus. We do this in our lab 7 times in a row to make sure the association is really getting through to the animal. And then what we can do is on another day, put the animal in the box, change the box a little bit so it doesn't necessarily know it's in the exact same place, and play that tone again and ask the animal, what does this tone mean to you? And so this is a paradigm that's been around for decades, and what this paradigm has kind of been distilled to in terms of whether we determine that the animal has learned that association is we ask, is it freezing or not? And so this is an example of an animal, a rodent in a freezing posture. You've probably seen this. It's kind of deer in the headlights looking. And basically every fear conditioning experiment that's ever been published, thousands at this point, the data are, is it freezing or not? Or how much time does the animal spend freezing? But there's lots of different things animals can do to show that they are afraid. And so that's why the title of my talk is What Have We Been Missing? Because we've essentially ruled out anything that isn't freezing. And part of the reason that has maybe gone away is that freezing is very easy to look at, right? So here's a person, he's freezing, he's not doing much else. But when a person or an animal is in a threatening situation, there are some other things that they might do as well. So for example, you might try to confront the threat, or you might simply just try to run away and escape. And I think that some of the reasons that we don't see publications where they're looking at some of these non-freezing behaviors is that by and large fear conditioning work has been conducted in male animals only. So what you're looking at here is a chart of basically a giant PubMed search. So if you search PubMed for fear conditioning papers and ask what is the sex of the animals in the that were used in the experiment, you can see that over the last 20 years, this has really been predominantly done in male animals only. You can see there's a little bit more of both as we've gotten to more recent years. Female only papers almost, it's really just a handful every year. And so I think a part of our lack of appreciating the sort of behavioral diversity of what animals can do in a fear conditioning paradigm is due to this bias towards only studying male animals. Now, when I first started the lab, I ran a really big experiment with, or I should say I ran it. My grad student ran it. Her name is Tina Gruna, and she was watching these videos and she had lots and lots of males and lots of females because we were really looking specifically for animals that just naturally behave differently in fear conditioning. And she said, this video again. She's like, I'm watching these videos and I'm trying to score whether or not they're freezing. And some of them, the tone comes on and you can see them dart across the cage like this. And, we're behavioral neuroscientists. It's hard to watch a video like this and say, well, I don't think the animal is afraid, or I don't think the animal learned what that tone means, because it's clearly responding to the tone. It's just not freezing. But if all you were doing was measuring freezing behavior, this animal would get a low score, and therefore you would interpret it as not being afraid. But she was like, I think, I've seen this enough now where I think this is something real and I want to keep studying it. And so I said, okay. And so essentially what she did was try and understand what this behavior was, when we saw it, what animals did we see it in. And so in order to do that, the first thing she had to do was define this behavior, define what is a dart and how do we see it. And so just to give you these definitions right up front, a dart she defined as a discrete locomotor response that happens during the conditioned stimulus, so during that tone presentation, that reaches at least 20 centimeters per second. This was sort of the velocity threshold for darting versus, just hanging out. And a darter, so these are animals that engage in darting behavior. They do it at least once later than the first two tone presentations. So we don't want to count darting as an animal that might just be sort of, startled by the tone coming on or anything like that. We want to only count it if they do it sort of as a learned response. So now that we have these definitions, she could go in and say, okay, how many animals are doing this and who are they? And what she found was that at a ratio of about 4:1, females were much more likely to engage in darting behavior compared to males. She found about 40% of females were classified as darters, whereas only 10% of males were. And we looked at a couple of things that were like low-hanging fruit as possible alternate explanations for this. So a lot of people might say, oh, well, the females weigh a lot less than the males do. They're lighter on their feet, so maybe they're just more easily, engaged in this active response. Doesn't really seem to be the case that the animal's weight is contributing to their propensity to dart. We also had estrous cycle data for these animals, and so we said, well, is it only females that are doing it in one estrus phase or another? Also doesn't seem to be the case. We saw darters in all 4 estrus phases, as well as non-darters in all 4 estrus phases. So doesn't seem to be anything related to those particular factors. Then what she did is she said, okay, let's separate out the darters and non-darters. Because she had this really huge cohort, she had the statistical power to do this. And she said, let's look at some other things that are happening during fear conditioning. So one thing she looked at was the shock response. So this is essentially how fast do they jump when they get that foot shot. And what she found was that across the 7 trials, animals that had been classified as darters, they start to escalate their shock response. So in other words, as the shocks progress, they start responding with bigger and bigger movements. Whereas animals that did not get classified as darters, they sort of kept steady. They had sort of the same shock response the entire time. If you look at freezing behavior, you see that probably, as you would expect, the animals that were classified as darters were freezing less. And that is in part because they are darting instead. But what you can see is on the next day when we do an extinction test, they end up sort of at the same place, and they extinguish at essentially the same rate. What was interesting is actually on the 3rd day when we played that tone to test their extinction, animals that were darters froze less than the animals that were not, suggesting that there is something about darting 2 days prior that is sort of priming these animals to learn extinction better and to hold on to that extinction. So we think that maybe having this kind of like behavioral flexibility might be contributing to that, their ability to learn extinction. All right, so one of the other interesting things that we started to look at is, again, going back to most fear conditioning papers, the only data you get are from that period where the where the tone is actually playing. Even though the whole paradigm is going on for maybe half an hour, 45 minutes, there's a whole lot of other behaviors that you're just tossing aside that also could be telling us about the way the animals are learning. What is their psychological state? Are they afraid, etc. So we looked not just at the tone epoch, but we identified some other epochs that we thought were really important and interesting too. And so these are periods of time before the tone. So that's what we're calling pre-tone. The period of time during the shock or immediately surrounding the shock itself, as well as a post-shock period to see what the animal does kind of in the semi-immediate aftermath of each shock. And so in order to be able to capture all of those behaviors during each of those time points, an undergrad in my lab, Anna Lee, actually developed a new Python program that would allow us to get all that information. So we took all our videos from the fear conditioning experiments, ran them through EcoVision, which is a locomotor tracking software that will give you velocity data for every single frame of your experiment. And then what SecurityRat does is it takes that raw data, processes it into these epochs that we're interested in. And so what you end up getting here from running your data through Scaredy Rat is a picture of what's going on for each animal across the entire fear conditioning session. And so what you can see here, this is just velocity on the y-axis and time on the x-axis. And I think one of the easiest things to identify is each of these big purple lines, well, the colors are coming out, but every tallest line, that's when the animal is getting shocked, as you might imagine. So very, very easy to distinguish those. What's happening during the turquoise periods, that's when the tone is playing. And so what you can see is that early on in fear conditioning, the tone doesn't cause any kind of response. The animal is basically just doing this, same locomotion velocities that it does during exploratory behavior. As it starts to get shocked and put together, like, oh, this is kind of the bad place, you start to see the velocity go down, and that is due to freezing. And you can see here in the middle, the animals are basically just entirely still for the whole time until they get shocked. Where things get interesting is when the animal switches into darting behavior. And so you can see that with these now elevated velocities during that tone presentation, that is the animal beginning to dart. So what's really interesting about this is that animals that become darters, they don't start out darting, right? They start out on kind of a normal trajectory where they're moving around, the tones and the shocks start to happen, they start freezing, but something then just causes a switch. And this is something that's been very interesting to us because we want to know what is causing the switch. Now, why does darting happen? And so one of the things that we've now spent the last 5 or 6 years trying to explore and understand is if darting is a conditioned response, and we believe it is it sensitive to the parameters of the experiment? Meaning if we changed things like the intensity of the shock or the amount of space the animal has or the number of trials, are we going to see changes in darting? Is there any way we can get more males to dart, or are they really just never going to dart? They don't want to engage in this behavior. And are there other behaviors that we can capture in a fear conditioning paradigm that might indicate the animal's psychological state? Because it's clear at this point that freezing by itself is not enough. So all of that work was published last summer in a paper spearheaded by my PhD student, Julia Mitchell, with the help of some really excellent undergrads in the lab. And I'm putting a QR code here because a lot of universities, including my own, don't have access to the Learning and Memory Journal. So if you want the paper, this should get it for you. And just to give you a quick kind of super fast summary of what you're going to find in this, you find that if you increase the shock intensity, we actually see less darting, which seems a little bit paradoxical, and we're working on trying to find out why that's the case. If you do not have a conditioned stimulus, so in other words, if you basically just do a context conditioning experiment, you still see darting, there's no real change there. If we increase the size of the chamber, we see more darting, so the animals have more room to express themselves, they're going to try out other behaviors more readily. And if we extend a fear conditioning session, so give them 20 CSUS parents instead of 7, you see more animals, including males, will switch to that darting behavior. So all really interesting manipulations of the parameters of the experiment that will change how much darting we see. But because that's published, and I have so much cool new unpublished data, I'm not going to give you those data here, except for this one graph, which is from the paper, because this was something that was so reliable in both males and females here, which is that shock response, which I showed you before from the first paper, we see that really reliably. Animals that ultimately become classified as darters, you see that they elevate their shock response, sometimes even before the darting happens. So this shock response seems to be a predictor of who is going to become a darter. And so what you can see here, this is shock response. So exactly how fast they move when they get shocked. And this is post-shock activity. So that 30 seconds period after getting shocked, they continue to locomote faster than animals that are never engage in darting behavior. And some of these animals, they might only do one dart in the entire session, but that one dart as a criteria is enough to place them into this shock responsive category. So what this has told us is that essentially darting, now we can think of it as a conditioned response. Darting happens during the conditioned stimulus that is predicted by the magnitude of the animal's unconditioned response. So the unconditioned response, how they respond to that foot shock. And so this connection, I think, is very interesting. And so we've been thinking about this, and I think the most obvious question to ask for sort of hypothesis about this is darters are simply more sensitive to pain. And that's what's going to cause them to have this heightened shock response, as well as engage in the darting behavior that we see for their condition responding. So we've been working on different ways to try and ask if this is the case. And so one way of doing that is by asking, well, what do they say when they get shocked? Do they say, ouch, more? And the way that we've been doing that is through recording their ultrasonic vocalizations. So this is a project that has been led by my postdoc, Pavlovian, who moved here from Finland in the middle of the pandemic, set us all up, got a paper out in like a year and a half, just absolutely amazing. So ultrasonic vocalizations, obviously we cannot hear them, but there are fancy microphones you can buy that can record these. And then there is a really nice machine learning based tool called Deep Squeak, which will help you analyze what that data is like. So there are two main kinds of USBs that the animals will make. We are calling them shock calls, which look like this. And you can see there's like a lot going on. They're very, very noisy when they get shocked, as well as alarm calls, which are not made directly in response to the shock, but they make them throughout the air conditioning session once they figure out that they're not having fun. So these are these kind of low frequency 22 kilohertz alarm calls. They're very long and extended. I do not have sound for you, but they're very sad. They're very sad sounding. So what we can do then is measure these different kinds of calls, see how they track both with just general sex differences, as well as whether they relate to darting at all. So what Michaela did was she trained rats in fear conditioning in one of three groups. They either got a 0.3 milliamp foot shock, 0.5 milliamp foot shock, or a 1 milliamp foot shock, as a way of seeing kind of how all of these things scale, and understanding again a little bit more, having a little more control or information about the amount of pain the animals might be. So first, what you're looking at here, this is just that sort of locomotor shock response, and you can see that in both males and females, we get a sort of scaling with the amount of the intensity of the foot shock itself. This is higher in females than it is in males, essentially across the board, which we've seen in the past. When we look at how much they scream when they get shocked, you can see that this also scales with the intensity of the foot shock. So this is a good indication that the animals can discriminate between these three different foot shock levels. And here we didn't see any sex differences. Yeah, oops, a little messed up. Animations there, that's okay. So next we said, okay, are the darters different from the non-darters? So we asked how many darters do we have, and you can see in these pie charts here, the males have about 7% of them were categorized as darters, and females about 22%. So a little bit lower than we've seen before, but enough to do some analyses. And again, we looked at the shock response. Again, replicated our previous finding that darters have higher shock responses than non-darters, and this is true for both males and females. But they don't make more noise when they get shocked. So this is an interesting kind of discrepancy here that is maybe putting a little bit of a sort of wrench in our idea that the darters are simply in more pain than the non-darters. If they are, it's only reflected in the way that they behaviorally respond to the pain, locomotor, but not what they do vocally. We also, just to give you a little bit more data from these, we see that the alarm calling latency, so now we're talking about alarm calls, not shock calls, these also are going to scale with shock intensity, but this is much more true for the males than for the females. So this is essentially how long it takes, how long the animal is sitting, getting fear conditions before it starts making these alarm calls. And you can see that when the shock is the highest, they start making the alarm calls sooner than when the shock is not so bad. So the alarm calling scales very nicely, especially in the males, a little bit in the females, but not quite as beautifully. When we look at the total number of alarm calls that the animals make, this is also much more present in the males than in the females, and overall we have a huge sex difference in the number of alarm calls the animals make. If you look at the alarm calling as a function of the conditioned stimulus itself, you can see again that in the males, it's just beautiful how things really separate, and you can see they kind of ramp up pretty quickly to whatever their plateau is going to be for that shock intensity, and then they just stay there for the entire session. They just don't stop screaming. Whereas, and if you look at the next days, right, you can see that they do show alarm calling in response to the tone. So this seems to be a conditioned response. In the females, though, they don't follow that same pattern at all. It's like they try alarm calling and it's not working, so they decide they're going to save their breath. And again, very little responding on the second day when they hear the tone. So it just seems to be less of a conditioned response for the females. What was interesting is that some rats will never make alarm calls. They definitely are getting shocked. They will make those shock calls, but they just don't start making alarm calls. And so Michaela thought this was kind of interesting. Kind of in the same way we have darters and non-darters, we now have screamers and non-screamers. And there's a sex difference here where 75% of males will elicit alarm calls, whereas only 55% of females do. And we looked at whether those animals have differences in some of their other behaviors. And you can see that in the males, the screamers are also freezing a whole lot more than the non-screamers, whereas for the females, there's no relationship there between the alarm calling and the amount of freezing that they do. So this is a really, really robust sort of sex by behavior interaction here, where there are things that go together in the males that just do not seem to relate to each other in the females at all. So what did we learn by listening to our rats? We found that ultrasonic vocalizations in males will scale with shock intensity and freezing, but not so much in females. Females make fewer USQs overall, and they don't really track as well with other behaviors. And we also know that darters did not make more shock calls. And so there may be this dissociation between sort of the modality of shock responding. So in other words, the females especially might be more prone to the locomotor related responding to getting shocked, whereas the males might be more inclined to start eliciting these alarm calls. And here is the QR code for this paper. This is an eNeuro, so everybody has access. And Michaela just did an immense amount of work for this project, and she looked at basically ultrasonic vocalizations across the board through extinction. She did an unpaired experiment to see whether or not it was in fact reflective of associative learning. And she did all these really beautiful correlation and matrices where the kind of end result of all of that was that a lot of things related to ultrasonic vocalizations are related to each other and related to freezing in males, and nothing lines up in the females with this particular metric. So this was pretty interesting to us, and I think most importantly, starting to put some doubt into our heads as to whether what we're seeing with the shock responding in the females is in fact pain response. But we did not give up. And so another way to look at this that Julia asked was, will darters show sensitivity to foot pain in other pain modalities? And so what she did was she gave the animals what's called the hot plate test. And in case you're not familiar with this, basically put an animal in a glass cylinder, put that on hot plate that's tuned to a pretty hot temperature, and wait and see how long it takes the animal to pick up its foot and kind of start licking its foot because it hurts. And as soon as that happens, take the animal out. So it doesn't take very long. It's usually just less than a minute. And then we can record that latency as a measure of how sensitive these animals are to foot pain, where if you have a long latency, you would say they were less sensitive. So she gave them a hot plate test and then put them through fear conditioning, find the darters, look at the relationships between behaviors in the hot plate test and behaviors in fear conditioning. And so first we looked at just basic sex differences, and we found that females have longer latencies than males. This is in line with what has been seen by other labs, and I think the general idea here is that this is due in part to the males weighing more, so they have, sort of heavier on the hot plate, so they're going to sense it sooner. But what you can see here is our darters are noted in triangles. They did not seem to, nothing really lined up with their foot shock or their foot sensitivity. However, we then looked at some other things with some correlations. And what we found is that if we looked at their freezing, conditioned freezing during fear conditioning, correlated with their latency to withdraw. These are all the females here, nothing too spectacular, but I will show you our darters. They're now in blue. And so you can see the darters are doing what we think they're doing as far as freezing goes, freezing less. But if we take the darters out, suddenly this amazing correlation comes in. And so it seems that for non-darters, there is actually a very significant relationship between their hot plate sensitivity and their freezing behavior. The same exact thing happened when we looked at that shock response. So here again, here's all our females. There are our darters right up at the top of the shock response sort of spectrum where we would expect them to be. Again, take them out and suddenly there is a beautiful relationship between hot plate sensitivity and foot shock sensitivity. And this is in the direction that you would expect it to be. So animals with the highest response to foot shock have the shortest latency to withdraw their feet in the hot plate. So that's females. In the males, we saw a little bit of a different story. So here are our males. This is freezing correlated with latency to withdraw. We do have a couple of male darters here, but taking them out doesn't do anything to that relationship. If we look at shock response, again, here's our darters way up here. So they're looking like darters. And here taking them out does actually give us a significant correlation, but it is in the opposite direction from what we saw in the female. So in this case, higher sensitivity in hot plate test predicted lower sensitivity in the shock response. So that was kind of surprising, and we are not entirely sure. Okay, so what we seem to be getting to here is the darters, especially in this hot plate test, are really just marching to their own drum. They are not following the normal rules of what most of the other rats are doing. And so we are becoming really interested in identifying what is really going on in these animals. And again, the thing that's continually remarkable to me is that they just have to dart once. And it's like, that's it. They're on their own plane. And speaking of individuality and how important that is, I just wanted to throw up this paper, which I worked on with Bob Data over at the Harvard Med School. And so this paper, essentially what they did, they were asking, are there reliable behaviors that can be attributed to different phases of the estrous cycle? And what Bob has done is he developed this machine learning tool called motion sequencing, or MOSI, that can distill animal behavior in 3 dimensions across time into tiny little syllables and identify the patterns of behavior. And what they found was that there are no reliable patterns of behavior that are associated with the estrous phase. The thing that really pops out in this study is that each animal essentially has its own personality, and that behavior is reliable across time, much more so than any kind of sort of group level estrous phase behavior. And so these are just cluster analyses showing how different behaviors cluster together. And on the left, they are clustered by estrous phase, and you can see that everything is kind of all over the place, or they're color-coded by estrous phase. On the right, they're color-coded by animal, and you can see that each animal's behaviors are very much clustered together, suggesting that really identifying these individual differences in animals is what might really give us some insight into what is going on potentially in fear conditioning as well. All right, so we have, that was a lot of behavior. So I'm gonna do a little bit of brain stuff too. So one of the things we've been asking is, are there sex differences in the brain regions that get activated or that are driving behavior during fear conditioning? And so one of the ways we've been testing that is through a couple of different brain regions that have been stained for CFOS, a marker of neural activity. And I'm going to show you amygdala data today, since that is sort of primary brain region that we often associate with fear conditioning. So this was done by an undergrad in the lab, Mary Claire Pikus, and all Mary Claire did was sacrifice animals after fear conditioning that either went through 1 milliamp foot shock, 0.3 milliamp foot shocks, or just heard the conditioned stimulus alone, and then counted CFOS content in central amygdala, basolateral amygdala, and lateral amygdala. And so I'm going to show you central amygdala data first, so that you believe the rest of the data. As you can see, in both sexes, CFOS scales really nicely with the shock intensity. You can see it goes up from baseline in the 0.3, and then a lot more at 1.0. Same thing in the females, although overall females have much less CFOS than the males do. When we look at the VLA, we start to see that, okay, this is still the same kind of trend for the males, goes up with more shocks they get, makes sense. And the females, not so much. This seems to be pretty blunted. And when we look at the lateral amygdala, this is the case even more. So you can see again, lateral amygdala, beautiful scaling with shock intensity in the males, and just no response at all in the females compared to the animals that were not getting shocked. And the part of the reason this was so striking to us is that the lateral amygdala is where fear conditioning is supposed to happen. You know, we've been learning for decades that the information, the somatosensory information about the tone and about the foot shock coming from those sensory thalamus and thalami, and having that first synapse in the lateral amygdala. This is an image from a review paper that's at least a decade old at this point, and about how sort of the fundamental fear conditioning circuitry works. Information comes in from sensory cortex, sensory thalamus, the lateral amygdala, that communicates to the central amygdala, which is the output structure of the amygdala, and that's going to drive all the behavioral responses that you see. This might not be true in females, and to my knowledge, no one has really done this kind of sex differences work before to really track what is happening in these different brain regions. And all of the dogma we have about neural circuitry that is related to fear conditioning all comes from male animals. And so it might just be that these pathways are different in females, and based on our CFOS data, what it looks like is happening is that everything is kind of just going straight to the central amygdala in females. So that's a hypothesis that we need to test, and we are working on designing the experiments to do that. To switch to a different circuit, we have also started looking at projections between prefrontal cortex and the periaqueductal gray. The reason that we focused on this area is that periaqueductal gray, or PAG, has been known for a long time to be organized into these really cool longitudinal columns that are functionally distinct. And the basic division of labor here is that the dorsal PAG, or DPAG, controls escape behavior. So if you electrically stimulate the DPAG, animals start running around. The ventral PAG is in charge of controlling freezing behavior. And so the PAG then is this really beautiful target for potential switching of behaviors from something like freezing to darting. And so we were really interested in that because we believe that the switch to darting is, in some senses, a choice that the animal makes. We think it's going to be controlled by sort of upstream, more executive function kind of areas like the prefrontal cortex. And the pathways from the PFC to the PAG have been mapped, and were actually the subject of a paper that I read for my qualifying exam when I was in grad school. And once we started thinking and looking at darting in the lab, I was like, I know exactly what paper we need to read. And so we went back to this paper, which has since been kind of brought back into the spotlight by a couple of different people. The basic organization of the circuits, according to this paper, which is only in males, I will say, is that the ventral PAG receives input from the ventral and rostral parts of the prefrontal cortex, which are shown here in blue, and the dorsal PAG receives inputs primarily from dorsal and caudal PFC. And so our thought was, okay, if we stimulate this pathway that goes to the dorsal PAG, so from basically caudal PFC, especially infralimbic cortex, we're really interested in because of the relationship with extinction that we saw in the first paper, maybe we can see more darting. So what Julia did was she used chemogenetics or DREADS techniques in order to isolate this circuit and stimulate it specifically. So she put an avian virus carrying Cre kinase that's going to go retrogradely back from the dorsal PAG, infect cells in the PFC, and then she puts Cre-dependent DREADS carrying virus into the PFC. So now we've got our circuit isolated. She can give CNO now to activate that the DREAD, and now the pathway is active during fear conditioning. So she here's some beautiful images of her labeled cells showing that the viruses went where they were supposed to. And then she fear conditioned the animal. She put them through extinction as well as an extinction retention test. And so Interestingly, we actually did not see more darters, and in the females, we actually saw a little bit of a reduction in that shock response, which was surprising. But when she looked at fear conditioning, what really stood out here is that, excuse me, she saw that improved extinction retention in the animals that had that infralimbic to dorsal PAG circuit activated during fear conditioning. So again, this is a delayed effect that seems to kind of mimic what we were seeing in darters, which is that their extinction retention was better, even though nothing was different in the extinction itself, or even during fear conditioning. So it's almost like she kind of recreated the darting state of mind without actually inducing any darting per se. In the males, when she activated the circuit, there was no effect at all. We saw actually a couple more darters than we normally do for a cohort of males, but that was our only potentially interesting thing for males. What this is telling us, though, is that some of these textbook circuits, so the image I showed you showing the connections between the PFC and the PAG, most recently are from a textbook called like the rat brain, and that's the whole thing. But of course, everything, we know all of these neuroanatomical studies were done primarily in male animals. And so it may be the case that some of these textbook circuits are actually not so textbooky, or the textbooks are only good for males. One of the other brain regions that we've been interested in is, again, the ventral PAG, which controls freezing, because it's also, of course, a possibility that it's not just that the dorsal PAG is driving darting, but that you need an inhibition of the freezing part of the PAG as well. So we wanted to say, okay, where is the ventral PAG getting its information from? So we put another retrograde virus in the ventral PAG. This is just expressing GFP, so we can see where in the PFC is projecting to the BPAG. And what you can see here in these images of the PFC, so these are the labeled neurons that we know are going to the BPAG. The labeling in males and females is really, really different. If you look up here at the anterior cingulate, in males, there is very little labeling, and this is what was predicted based on the textbook, that the anterior cingulate does not project to BPAG because it is dorsal. So they say ACC only goes to dorsal PAG, but you can see, of course, that there's tons of labeling in the ACC in the females. We see another difference here in the infralimbic cortex, where the males have lots of labeling in the infralimbic cortex, and the females have very, very sparse labeling. So these two sets of projections from ACC and IL down to the BPAG could be a target for why we have sex differences in fear conditioning behavior. And so we're doing some of that now. We've started using fibrophotometry to record from these projections, so we can see what's happening in the animals as they're going through fear conditioning. And this project is being taken over by a new PhD student in the lab, Dr. Shepherd. Okay, so here we have our conclusion slide. So just to kind of sum up, I know I showed you a ton of data, but what you can see from all of this is that by examining multiple behavioral metrics in Pavlovian fear conditioning, we can see different kinds of modalities in which sex differences can emerge. So if you're looking at locomotor behaviors, you might see new things in females. If you're looking at ultrasonic vocalizations, you might get some new information in males. It's really important to consider that different behaviors excuse me, can have sex-dependent purposes, right? So the differences in ultrasonic vocalizations is really interesting and surprising to us, but we have to ask ourselves, well, what is the purpose of a USB when the animal is alone in a box? And it may be that for males, it makes more sense to try and communicate. For the females, maybe it doesn't. And same thing goes for escape versus freezing behavior when a threat is detected. One of the big things I think we can see here is that sex differences in neural circuit organization are likely playing a role in mediating some of these behavioral differences, and we really can't assume that whatever has been established in males is going to definitely translate to females. They're, clearly the females are learning, but they might be recruiting different brain regions, different circuits in order to do that. And I think overall, by identifying sources of variability in complex behavior profiles, it really can give us new insight into the different ways that the brain can learn. And so I know, scientists want our data to be super clean and tidy, but the reality is that variability in your data is telling you different ways that things can happen in the brain, different ways the brain can work. And I think that that's worth paying attention to. So that I will just say thank you and thank my amazing lab of mostly undergrads and grad students, and I'm happy to take any questions.
Everyone, we have questions.
That was awesome. So I was curious whether you looked at differences across age as well, and potentially whether like females have had a chance to meet yet or not, and whether that changes things. I study flies, but flies, like virgin females tend to appear in some of this males, and then once they meet the change, so I'm curious.
Yeah, so we haven't done any age-related studies, but we did just do a couple of experiments looking at the effect of maternal reproductive experience as well as maternal experience, and we found some really, really interesting findings, which is that the animals that had given birth as a group had less freezing than the virgins, but that effect was entirely driven by darters. So there was something very interesting darter by pregnancy interactions going on there. And we just got those data, which is why I haven't had enough time to think about it even, but it was very striking. So we're really excited to follow up on
Okay, hopefully this one's working better.
Okay, so my question is about this is fascinating, by the way. The connection between the prefrontal cortex and periapical gray between males and females. I'm curious, is that an all female thing? Is it a darting female thing, or is it the magnitude any different in the darter?
So that is A1 of the R21 that we got this year. So yeah, so basically the images that I showed you, that was just like a tiny little pilot, but it was so striking. So those animals were not behaviorally profiled. So they were just, males and females who grew the virus. And so what Viv is doing now is exactly that experiment. So she's doing the injections, letting the tracer go, behaviorally characterizing the animals, and then we're gonna count all the cells and say, like, do darters have more or less, or things like that.
So did you look at weight or the size of the rats with dartings, like with connected foot shot? Did you look at the factor that they play in chance in which all the electric was all bars electrified, or was this like a safety with the practical?
So we did look at the weight. I had that slide in the beginning. So there was no relationship between the weight of the animal and whether they became a darter or not. All of the definitely all the animals get foot shocks. The bar is called like scrambled. So as long as they have 2 feet on the grid, then they will get shocked. But it's very hard for them not. There's no space.
Okay,
Yeah.
that's interesting. This stress is safe with the foot shots and become like size. The older the larger the rat is, they only respond, they have a higher sensitivity, a less sensitive to that. That's interesting.
Okay.
Thank you.
Hi, thanks for the great talk. This is from a non-expert in both rats and air conditioning. But do you know whether or not other types of responses, I'm thinking of an acoustic, I think play a tone and then a loud tone to make the animal startle, whether or not you would see darting and under paradigms like that, whether there would be sex differences? Has that been looked at?
So I believe that there are sex differences in acoustic startle responding. Usually for those, the way the chambers are designed for that, the animals are pretty contained, so you wouldn't be able to capture any other behaviors besides just like, do they startle or not. We have not yet done the experiment to say, do darters have a higher startle response, but we do, our colleague Heather Brenthouse down the hall has the chamber, so it's a, yeah, we should do it.
Can you explain gender and environmental on why
So do you mean sort of like what is the ecological reason for darting versus freezing? I don't want to speculate too much because it's really not my background. I think it could make sense that for males, freezing makes more sense, just try not be detected, maybe because they're just not that fast of runners. Although we know that when we can create male darters, they're just as fast as the females, and so that's probably not it. But for the females, it might just be like, you know what, I'm just getting out of here, as opposed to trying to just evade detection and wait out the predator. So it's a good question. I don't know if we have a good way to test that empirically in my lab.
Really nice presentation. My question is kind of similar to that one, which is, what do you think what do you think the darting represents, or its potential function? Is it perhaps a furtive escape response, even though there's nowhere to go? Do you think that previous experience with escape would change the frequency of darters, because they will have experience that you can run somewhere and get away, even though there's nowhere to run in those chambers?
Yeah, so great question. To me, it looks like an escape response, like they're trying to find a way out, even though there isn't one. It is, in most cases, their first day in the box, and so they might think there's something that they could, a secret trapdoor or something. But I don't know off the top of my head. We did, in the paper in Learning and Memory, we did try doing that sort of second-day test in an open field, and you do get more darting when there's a bigger space. And so that indicates that, yeah, they're probably just like looking for a way out.
There's one final question from the upstairs
Oh.
screening area. This is a question from upstairs. So I'm receiving live via text. So how consistent are the darting strategies? For example, like in other words, will a darter revert to a non-darter or vice versa? And how malleable are responses over time? Can a freezer become a darter?
So all great questions. It's hard to test the way that we have things set up, just because you can't really fear condition an animal more than once, because it's about like what's happening across time. So what we do see is that animals, and I know I've said this a bunch of times, but like they do really only have to dart once. And so that means that on other trials, so they might dart on, say, trial 5, on trial 6 and 7, you're going to see some freezing even in the darters. It's not like once you dart, you suddenly are never going to freeze again. So there is some, kind of intermingling of the behavioral strategies. The one thing that does seem to be very reliable is that shock response. So that, and I showed you the graph. I mean, it's like once they get up there, they don't ever stop responding at that level.
All right, well, thank you again.